This would be done by me. Then you would have a physics period done by Upamanyu sir. Okay. And then the third period is chemistry, where you'll have your regular teacher. And then the fourth period will again be physics. Mr. Praveen will handle it. Okay. Your maths teacher has gone home. Okay. He will be resuming classes after 2 days. Okay. So there is no English class. So these are the timetable for your for seeker students. Okay. Here this is common for all the things. I'm not sure if KNMS is having English or not. I'm not sure about it. Okay, I'm not clear about those things. Please refrain asking questions about it to me. So this is the schedule for the day, and then biology students will have a test between 5 o'clock to 8 o'clock. The test window will be open during this time. So bio test will happen between 5 o'clock to 8 o'clock. The questions will be available online at stg.learnity.com. You have to log in with your user ID password, and then you will be able to get it. I guess you guys should be knowing the portions. If you are not clear about it, I am sorry about it. You have to listen to the class properly. I cannot be repeating the same thing again and again and again. Okay. If you don't know the password, if students of EDUS are like having classes during that time, I think you can skip the test. I can arrange the test separately for you. Okay, so you can text this thing with your or Blossom, whatever it is. And then email ID. So these things I want you to provide me as a text either here, or you can also mail it to You quickly do this thing. I mean, like, do it before quickly. You can take it at any time between 5 o'clock to 8 o'clock. 5 o'clock to 8 o'clock, the link will be open for you. It will be available. You just log in, it will be available. You can take the test. Okay. It is only for your thing. So, students, first, please be very patient. Listen to me properly. So today bio test will happen between 5 o'clock to 8 o'clock. The window will be open during that time. You should have a user ID password for your stg.learnit.com, learnit password. If you're not having your stg.learnit password, what you should do, this is what you should do. You should send me a text either to my mail ID or to my WhatsApp ID. Okay, with the full name, batch, email ID and phone. I will not be open for any technical issues in Learnit. This is my number just to sort out the issue. Okay, so this will be done only for today. I don't generally respond to my number messages. If you want to write, you can write, no issues. You can contact Ramesh sir also. Yes, you can contact Ramesh sir. If you are like interested, you can reach out to Mr. Ramesh as well. If you got an ID few days back, it is fine. That is the ID that you have to use. I am the bio teacher. I know only about biology. I don't know about other subjects. Okay. So today's test is my duty. So I am taking care of it. You will be having 3-hour JEE test, 3-hour NEET test will happen from this week, most probably. Coming week, forthcoming week. Okay. How does it even matter, objective or subjective? Okay, so this is done. So today's work, my instruction is done. So let us start with the class. Okay. If you are having issues with Learnit, you have to use the Chrome tab. Okay, some of the instructions that you need to know about using Learnit. Use Google Chrome. The second one, the test is for only 1 hour. Yeah, right. It is for only 1 hour. But you can take it during that window, time window. You have to use Google Chrome. The second thing that you need to do is use incognito window. And the third that you should do is check your system time. And the fourth thing that you have to do is install the latest version. Okay, just in case if you are having any troubleshoot, these are the 4 steps that you have to try. Okay, only the error source will be only one of these four. There is no other possibility that you'll have an error. Okay, which means you have to do any of these things to get inside the thing. So if you are not able to get inside, this is the solution. Any one of them should be the solution. You should have used Google Chrome first of all. You should be using incognito window. That is the second thing. And then if you're not getting it, you have to install the latest version, and then you should check your system. Okay. The link will be available in stg.learnity.com. You just log in and you will have your test. Okay, so these are the instructions that you should be using. So in case if you have any difficulty, this 4 points should help you. Okay, so now if it is done, we can move on further. This is slide 1. Okay, children, I'm just getting this kind of questions which are very, very stupid. Okay, it's just showing your ignorance. There are people who are asking, is this test compulsory? Okay, now the onus is on you. So given that this kind of online classes are happening and then like everything is happening at a very remote case, I just think that it is your onus which plays an important role. Okay, if you want to study, you have to study. Okay, if you're skipping the test, it's totally perfectly fine because you have decided not to do it. Okay, and if you're like thinking that this is going to be like kind of an unwanted exercise, you can skip it. I don't have issues with it. Okay, people who are willing to work, we are interested to help. If you're not interested to work, I mean, like it's still fine. It's your choice. To get into something better, I think you should have intentions to do things. Vikash, please read the board, Vikash. Please don't ask stupid questions like these. Everything is there written in that thing. I mean, you people are testing the patience. So eukaryotic cell is what we are going to study. Okay. So the first thing that you need to study about eukaryotic membrane is eukaryotic cell is we are going to study about cell membrane. Okay, so you talk about cell membrane. What is cell membrane? Cell membrane is the otherwise called as plasma membrane. Okay, so normally whenever you're talking about eukaryotic cell will have plasma membrane. So this plasma membrane is a phospholipid bilayer. Okay, what is the meaning of phospholipid bilayer? It is made up of a combination of phosphate and lipids. Okay, so let us try to understand what is this phosphate and lipids. Please understand that lipids are fats. Okay, and phosphate group is there. So when phosphate group is attached with lipids, so when phosphate group is attached with lipids, what happens is there is this phosphate group which is there, which is attached with the lipid part. Now it is creating some sort of a structure where this part is becoming what we call it as hydrophilic head. Okay, so the word hydrophilic means hydro stands for water and philic stands for attraction. So basically it is going to be water along with the water attracting part, and then you have a part which is going to be lipid. This part is called as a hydrophilic tail. The word hydro indicates water and phobic indicates fearing. Okay, so basically it is almost similar to your measle. Okay, so there is a measle which is there formed in the soap. Okay, last in 10th standard you would have studied about measles. So it is like almost like missiles which are formed in your soap. So you have this particular structure. Your plasma membrane will be made up of a phospholipid bilayer. Okay. What is the meaning of a phospholipid bilayer? You just see here. This is a hydrophilic head. You'll have one more layer of hydrophilic head. Okay. In which you will have tails coming up like this. And you will have tails coming up like this. So your plasma membrane will look like this. It is made up of a bilayer. It almost resembles a sandwich. Okay, we call it as a sandwich layer also. Why? Because you see here, this is like resembling one bread and this is resembling another bread. The way you keep your bread in a sandwich, it is resembling that thing. So why is this kind of arrangement very important? Because this part which is there will be able to interact with water. Okay, it will be able to interact with water. Okay, which means it will not allow watery substances to come through this thing beyond this point. Because like water can enter till this particular point. But after that, this is becoming a hydrophobic part. So which means water cannot enter into the cell just like that. Okay. And similarly, if you're talking about fatty substances cannot come here only. It cannot come beyond this point only. Which means what is the main function of this plasma membrane? This plasma membrane is going to selectively allow substances. Okay. So this is like kind of a compound wall for the entire cell. What are the substances which can come into the cell? The substances which can come into a cell will be either fat soluble or water soluble. This kind of a setup is preventing both the substances to stay outside, which means water substances cannot also come inside the cell. The fatty substances cannot also come inside the cell. Okay, so basically it is like not allowing any substances to come inside. This lipid bilayer model is helping in such a way. Okay, so if you're going to see this lipid bilayer, you will be able to see that you will see this layer will be there. Excuse me. Okay, and then you will see something called as a transmembrane protein. So this is how your plasma membrane will look like. Okay, and then you will have something called as a you are able to see some proteins here. This is called as a membrane spanning protein. And then you have a transmembrane protein. Okay, your plasma membrane is almost like a compound gate. I mean compounded wall. Okay, so if you're going to talk about a cell will be having what? It will be like kind of like fully covered with plasma membrane. But you need substances to go inside and come outside. So that is where your membrane spanning proteins and transmembrane proteins are coming into the picture. Usually the transmembrane proteins are like kind of gateways, which will be seen in between, through which substances can move inside and outside. So basically this transmembrane protein, what is the meaning of transmembrane protein? A protein which will help in transfer in membrane. It is like almost like your gate in your compound wall. So what will happen is this is through this particular transmembrane protein, the substances can come inside or go outside. So these proteins are going to help in transport of substances. Okay, only it will not allow anyone just like that. These proteins will be like kind of helping only the substances that the cell wants. So only substances which can be allowed and permitted to get inside the cell will enter into the cell, and those substances which are not permitted or allowed will be like trying to staying outside. Okay, so the transmembrane protein is responsible for the transport. Okay, and plasma membrane is also performing functions of your signaling. Okay, so this, what is the role of this membrane spanning protein? Say for example, there are some molecules which are like hormones. Let us talk about insulin. Or let us talk about like say adrenaline. You guys know what is adrenaline. Okay. Adrenaline is released whenever we are there in an emergency situation. Okay. Whenever there is an emergency situation, when there is like some kind of like you're going in a road and suddenly there is some kind of lorry which is going to come and hit you, you either have to run away from that particular pathway or you have to like succumb to the death. Okay, either of these two things will be happening. So, but whenever your body is in the fight or flight mode, naturally your body will be releasing adrenaline. When adrenaline is released, your entire body should be functioning. So say for example, all the energy should be going to your leg cells, and suddenly all of a sudden the muscle cells of the leg should be moving. Which means in such a case and all, what will happen? Adrenaline is produced in very small quantities. Okay, what will happen is these adrenaline kind of molecules will, if it wants to make this particular cell work, what it will try to do is it will try to attach with one of those membrane spanning proteins. So let us say this is a membrane spanning protein somewhere there. So how will this adrenaline will make the cell work? It will come and attach with this thing. So the attachment of an adrenaline molecule here in this membrane spanning protein will send some kind of signals inside the cells. And it will make the cell work. So basically what will happen the moment this is attached, some information is going to the nucleus. And what will happen? The cell will be functioning in a particular way. So ultimately what I am trying to tell you is the transmembrane protein, some of the transmembrane proteins are helping in signaling processes. Okay, so what is the role of this membrane spanning protein? This membrane spanning protein is involved in signaling. The transmembrane protein is involved in transport. So you imagine a case of a school compound wall. Okay, you cannot get outside through the compound wall. You can go only through the gate. The gate is equivalent to transmembrane protein. You have a watchman who's there outside who's having a phone connection. Whenever someone wants to do something inside, he will be reaching out to the watchman. The watchman will try to consult the cell, the center, the school head, and then what he would try to do is he would try to revert back to them. Okay, so that is the work of your membrane spanning protein. Your membrane spanning protein is more like a watchman who is going to connect your whatever information is coming from outside to the inside. Okay, so this is what is your entire system. Are we guys able to understand? It can be either chemical or electrical. So a hormone which is coming and attaching is a chemical signal. Okay, so are you guys able to understand this thing? I have introduced lots of new terms here. You please note it. One is what we call it as hydrophobic. Then I have introduced the word hydrophilic. I have introduced a word phospholipid. What is phospholipid? Phosphate plus lipid. And then I have introduced the word transmembrane. And then there is something called as a membrane spanning protein. Okay, so you are able to see that transmembrane protein will connect two sides. It is open inside also and it is open outside also. But on the contrary, the membrane spanning proteins are attached only to one side. They are not kind of like communicating between the two things. So they will send impulses through different methods. So if you guys want to see this slide. Okay, so please understand that if in a phospholipid bilayer, almost 52% is proteins. And almost 48% is made up of lipids. So you understand that the phospholipid bilayer, 52% is proteins and 48% is lipids. Okay. Whatever we have studied here, what do you mean by extrinsic protein? Someone is asking, sir, are they extrinsic proteins, the membrane spanning proteins? I don't understand the word extrinsic. Can you explain? Okay, so someone is asking me that whether it means that the substance can. The word extrinsic means not outside. That is not the meaning of the word extrinsic. Okay, if you are asking me, looking at this diagram, it can be either intrinsic or extrinsic. It can be either inside or outside. Okay. If you are meaning the word, if you are considering that the word meaning of extrinsic it's outside, then yeah, you can say that it is not extrinsic. You can say that it is intrinsic and extrinsic, but intrinsic and extrinsic has very different meanings in English, which is like kind of I mean, I think you're using the word intrinsic and extrinsic very wrongly. Okay, crudely put, you can say that intrinsic and extrinsic means inside and outside. But intrinsic and extrinsic is like kind of different meaning. Uh, there are some substances which can pass through the membrane directly. Okay, so you have some low molecular weight lipids. Low molecular weight lipids are there. Some substances like alcohol. So these and all like can get inside the membrane without any kind of problem. Okay, but they are in very small quantity. So it is not only the majority, the bulk transport will happen through the transmembrane. But there are certain substances which can pass through the membrane just like that. Okay. So, but they will be like of very low molecular weight and it will not cause like much difficulty. So there are some bacteria which can come inside through also inside the cell without any difficulty. So it all depends on like how far the membrane is permitting. Okay. So we also have the membrane is not very intact because like microorganisms can still come in through that particular thing. It can make some part of the cells ineffective and it can still enter. It is not only the microorganisms. There are some minute chemical compounds which can enter, very small molecules. But usually the bulk transport will happen only through the membrane protein. Okay, I think I'm answering your question, Amreen. And then, sir, the phospholipid also has two tails also, sir. Yes, it will actually, the phospholipid will be like having two tails. I have drawn only one. This is usually like this. Okay, I was a bit lazy, so I did not draw. There is no use for alcohol inside the cell. It is just that alcohol is capable of entering. Alcohol doesn't have any use, but if a person is going to drink alcohol, naturally this alcohol will have to go something. Okay. Whenever I'm talking about an integral protein, what is the meaning of integral protein? The word integral, one more word is there. Most of these proteins are integral proteins. What is the meaning of integral proteins? It is integrated with the membrane. So let me show you a fluid mosaic model. This model is also called as a fluid mosaic model. Okay, so I'm just showing you some images. So let us try to see this picture. This is like kind of like I'm not sure if this is clear. Okay, I think it has become clear now. So you are able to see the structure. This is a membrane structure. You are able to see this blue color dots are what? What is this blue color dot? The blue color circle, what is that? We call them using dash head. What is the word that we will use? It is hydrophilic head. Very good. Okay. And then what is the tail-like structures which you are seeing? What do you call those tail structures as? Hydrophobic tails. Very good. Okay. And then these are all called as integral proteins. What is the meaning of integral protein? Something which is integrating two sides. The transmembrane proteins are called as integral proteins, which is integrated with the membrane, inside the membrane. Okay. So here you have like different types. These channel protein and then carrier protein and all, you don't have to learn it now. That is all quite advanced. Okay. And then you are able to see this is like the membrane spanning protein. Okay. So the membrane spanning proteins are there. Peripheral proteins are there. Those are all, this one, the peripheral protein and this glycoprotein which is there is all like what we call it as peripheral proteins. Whenever I'm using the word peripheral protein, it means it is lying in the periphery or the outer side or in the outer membrane. Okay. Whenever I'm using the word glycoprotein, it means it is a part which is having protein as well as carbohydrate. Here you are able to see this particular protein along with some chain kind of structures. This chain kind of structure is a carbohydrate. So it is a glycoprotein. Okay. So if you're talking about a cell, so this is how your membrane will look like in a cell surface. Okay, you're able to see the phospholipid bilayer. You're able to see the hydrophobic tail. So this is the inner surface of the membrane. This is the external surface of the membrane. Okay, you're able to see this glycolipid, glycoproteins and all are seen, which will be acting as membrane spanning proteins. Whatever these proteins which you are seeing here is the transmembrane protein or the integral protein. Okay, this model is what we call it as fluid mosaic model. And it was proposed by Singer and Nicholson. These are the two people. Okay, it is also called as a sandwich model. The plasma membrane is also called as a phospholipid bilayer. Yes, the channel proteins and carrier proteins are types of transmembrane proteins. Okay. So it was proposed in the year 1972. Okay, so now we need to understand one more thing. What is that one more thing that we need to understand in this concept is how transport is occurring and how plasma membrane is acting as a selectively permeable membrane. Okay, there are two types of transport which will be there. The first type of transport is what we call it as a passive transport. The second type of transport is what we call it as a active transport. The one which will not use energy, and active transport uses energy. What is the meaning of energy? We are talking about ATP. This is also ATP. Okay. So diffusion and osmosis are types of passive transports. Sorry, there was a small lapse in my side. Okay. So you have the cell. You are able to hear me now? Okay, thank you. So you have the cell which is there. Okay, suppose let us say that the water concentration here is more. Then naturally what will happen? The plasma membrane will allow the movement of water through some protein here. The concentration is more, the concentration is less. What will happen? Naturally the water will move inside the cell. This process is what we call it as diffusion. Diffusion is like kind of like what? Movement of any substance from higher to lower concentration. Okay, say for example, I'm using a perfume. Okay, I'm using a perfume and then I'm walking inside a classroom. The moment I'm walking inside the room, initially you will not feel the smell. Maybe like sometimes you will feel it. And then like, but after some time, what is happening is that smell is going to diffuse and then it is going to occupy the entire classroom. So how is that possible? Because like when the person entered, compared to that person entering time, and after like say 5-10 minutes of time, you will see that there is a change in terms of your the atmosphere there, and then like the fragrance there in that particular atmosphere. How is that possible? Because basically from higher concentration, which is that person who's there standing as a source, this particular thing has emanated into the class. It has gone into the class throughout. The molecules have gone. So that is what is diffusion. Basically, this place is like kind of like this person is having the source. From this particular person source, what has happened? That particular fragrance particles have emanated throughout the class. So it has moved throughout the class. How is it? It has moved throughout the class. It is because of a phenomenon which we call it as a diffusion. Okay. So like this, plasma membrane will also permit diffusion of molecules. Suppose if it is happening through the membrane, we call that as a osmosis. Okay, in plasma membrane, mostly osmosis is seen because plasma membrane itself is a permeable membrane, semi-permeable membrane. Okay, or we can call this as a quasi-permeable. The word quasi-permeable or semi-permeable means half permeable. Okay, so you will be able to see osmosis happening in your cell. What is osmosis? You just think about a situation. Let us say this you would have studied in 9th standard. Let us talk about a cell being kept inside the hypertonic solution. And then imagine a situation where it is kept in a isotonic solution. And imagine a cell being kept in a hypotonic solution. Okay, so now tell me what will happen. What will happen in hypertonic solution? In one of these particular cases, okay, let us say what is osmosis? Osmosis is movement of solvent particles from where to where? Higher to lower. Through what? Semi-permeable membrane. Okay, so if it is hypertonic, outside is having less amount of water and more amount of solutes. So what will happen? Water is more here and water is less here. What will happen here? Basically, from this particular part, it will try to come outside. We call that as plasmolysis. What is plasmolysis? Basically, the cell's protoplasm will start to shrink. It will break open. It will break. And then like what will happen? The cell will be like kind of broken. You just imagine a case of using a cucumber. Think of cucumber. Okay, you take cucumber, you cut it into small pieces, put it inside a tiffin box, just sprinkle some salt, close it and keep for some time. You open it, what will you see? After some time, half an hour or 45 minutes, you open it again. What will you see? You take cucumber, you're cutting it into small pieces, and then you're sprinkling some sprinkling some salt, and then you will leave it for 45 minutes, open it again. What will you see? Cucumber will not shrink and all. Cucumber will not be dry also. The tiffin box would become watery. Yes, exactly. So you will see that the entire tiffin box will become watery. Why is it watery? Because from water from the cucumber is going to come outside. Okay. The cucumber is not shrinking and all in this case. Like it is basically the water is coming outside. So this is what we call it as exosmosis or exocytosis. The protoplasm from inside the cell is moving outside. Okay, so the same thing, this is what water from the cucumber will come outside. So this is called as exosmosis. In case of isotonic solution, what is the meaning of it? The water concentration inside is equal to water concentration outside. So water will not move inside, water will not come outside, both will not happen. No change will be there. This condition is flaccid. The cell will not be happening, anything will happen to this particular cell. The cell will remain flaccid. In a hypotonic solution, the water concentration is outside is very high. The water concentration inside is low. So what will happen? The water from outside will enter inside the cell. Okay. And it will cause the bloating of the cell. The cell will get bloated. Oothi poradu. So it will become bigger and bigger, absorbing all water. It will become turgid. We call that phenomenon as what? Turgid. Okay. So when this is happening, what will happen? We call this process as endosmosis or endocytosis. Water is entering into the thing. Okay. We also call this a swelling. Yeah, you can say swelling is there. One more word. This is what is happening. So this is what is endocytosis or endoosmosis. The cell wall is not going to do anything. The cell wall will just make it. In case if it is a plant cell, no, the cell wall is present. Let us say that the plant cell has a cell wall and the plasma membrane is there. The water will enter. The plasma membrane will be like kind of bloating and then it will causing the turgidity. Okay. Beyond how much ever water is absorbed, the cell wall will be like kind of intact. Will not allow the cell to burst. But if the same thing is going to happen in an animal cell where there is only plasma membrane, the more and more getting accumulated, at one point the cell will burst. In plant cells, the bursting will not happen. It will not burst. In animal cell, it will burst. Okay, so that is the basic idea. So in case of hypertonic solution, if cell wall is there, nothing is going to happen. The cell wall will be intact, but the cell would have become shrunk. That's all. You have seen dried grapes and all, dried grapes or like dried fruits and all. So those are all losing water outside, but the cell is intact. You put it in water again, it will again bloat it. Plasma membrane is not over. I'll just give you some notes to remind you one more thing, one more time. In case if you don't have a user ID password, please reach out to me or please reach out to Mr. Ramesh. Anyone who's interested can take this test. If you're not taking this test, I'll just mark it as absent and then I'll pass it on to your school. Okay. So please understand that the test window will be open only between 5 p.m. to 8 p.m. So which means you have to take 1 hour of test. It is 45 questions. Yesterday night only I set the paper. So 45 questions, objective type test. You please write it and then like 1 hour you should be able to finish it off. Okay, so someone is asking a question. If the cell absorb water, then how concentration decreases in What do you mean by water concentration is not decreasing, man? The water concentration inside is low. உள்ளுக்குள்ளார கம்மியா இருக்கு. It is low. Outside it is more. So the water is entering till it will attain equilibrium. Okay. So inside the symbol here I am trying to tell is like water concentration inside the cell is low and water concentration outside the cell is high. So water will move from outside to inside. That is what I try to think. As soon as you complete the test, you will know the mark yourself. Okay. I don't have to correct the paper. As soon as immediately the test is over, you will know your mark. Yes, there are negative marking in this paper. All the instructions would be given. Please read the question paper properly. You don't ask me now. Okay, thank you, children. I don't know whether you have internal assessments and all or not. This is just for you to learn. If suppose it is required, they are asking, I'll give it. Okay, all the slides. Slide 1. Slide 2. Slide 3. Slide 4. Slide 5. Slide 6. Slide 7. Slide 8. Okay, thank you, children.